Assalamu alaikum students, how are you? Today we are diving into another side of metabolic regulations. In the last lecture you have learned about the insulin, the role of insulin, the metabolic roles of insulin, and usually insulin often takes the spotlight. The metabolic balance of the human body is a defined cue orchestration involving several players. And by the end of the session, you should be able to describe the synthesis and chemical structure of glucagon and somatostatin, um, explain their complex mechanism of action, and understand their metabolic index in both health and disease. So to understand these hormones, we must look at their birthplace, the lives of Langerhans, and we have already discussed in the previous lecture that the alpha cells, these are the alpha cells in the outer rim, and we secrete glucagon, and they are triggered by low blood glucose levels. While insulin, it is triggered by the high insulin levels and secreted by beta cells of the ides of the Langerhans, and these are the storers of the insulin, the beta cells, and glucagon has the opposite action as compared to the insulin. Then you have the delta cell, they are scattered in the rim, and they secrete somatostatin. This is the inhibitor, and the exocrine pancreas have acinar and duct cells surrounding the endocrine ides. So this is the ecosystem of and the ides of the Langerhans, and this is how the blood by the action of these cells, the blood glucose levels are being regulated. So this is the slide showing the metabolic tug of war, and it's showing the dynamic equilibrium. There is a constant tug of war between the insulin and glucagon and epinephrine. This insulin on the left side, it is pulling, and on the left side, while glucagon and epinephrine are pulling on the right side. And you can see that insulin is being released when there is a state of hyperglycemia. Okay, and it drives glycogenesis, lipogenesis, and ketolysis. So when this happens, these are the main metabolic functions of the insulin, and dominates when glucose is more than 120 milligrams per deciliter when there is hyperglycemia. In contrast to that, you can see the glucagon and epinephrine has the opposite action. And when the glucose drops below 70 milligrams per deciliter, there's a state of hypoglycemia, and in that time period, the glucagon takes the lead and driving glycogenolysis, gluconeogenesis, and ketogenesis. And our homeostatic target is to maintain a steady state between 80 to 120 milligrams per deciliter. So this is how the body's blood sugar levels are being maintained. So it prevents from hyperglycemia and hypoglycemia. All right. And here you can see this level you have to remember, because every time when you sit in the exam and the question is being asked that what is the normal blood glucose level, so most of the students they give another level, so you just memorize these levels and see when there is high blood glucose levels, it stimulates the glucose uptake by the cells, and pancreas release insulin, and by the insulin action, there is low blood glucose level, and when there is low blood glucose level, the glucagon action takes place, and liver releases the glucose, and pancreas release glucagon, and uh, then glucagon takes the action and stimulates glucose uptake by the cell. So this is how the health is not the static state, but a dynamic equilibrium managed constantly by these opposing forces. In this slide, you can look at the chemistry, and glucagon is the linear polypeptide consisting of 29 amino acids, and its molecular weight is 3,485, and it's produced as a massive 160 amino acid growth hormone by a single gene in the rough endoplasmic reticulum of alpha cells and leads to four peptides, and it includes the active 29 amino acids. So, after the formation of 160 amino acid prohormone, the final active hormone is left with only 29 amino acids, and they are being stored in the secretory vesicles and the alpha cytoplasm until triggered by the alpha cells cytoplasm until triggered by the hypoglycemia. And here you can see somatostatin, because we also have to study about it today. So it exists in two active forms. It has got 14 amino acid chain, and this is the 28 amino acid chain. And it is produced by alternative cleavage of single pre-protein, pre-protein. So this is the structure of somatostatin and glucagon. Okay. Now, how do these cells know when to release the glucagon and insulin? So, in the cells, high glucose enters via GLUT2, okay, and increase the ATP. I've already told you in the last lecture that GLUT2 transporters, then and that, when the glucose enters into the cell, it is converted to glucose 6 phosphate, glucose 6 phosphate. Then via glycolysis, it enters into the citric acid cycle, and there is oxidative phosphorylation, and it leads to the production of ATP into the mitochondria. And after that, there is when there is increased ATP, that closes the ATP potassium gated channels, and then it causes the depolarization. And after that, the voltage gated calcium channel open after depolarization, and then that insulin secreting the vesicles that secrete that has insulin inside it, by the action of calcium, by uh, with the incorporation of calcium with it, the insulin granules are being secreted by the exocytosis in the blood circulation. So this is how the high glucose response we get by the high glucose levels, and this is due to the spike in the intracellular calcium, the final stage of insulin release. Okay, and then you can see that this is the low glucose response, and uh, this is the hypoglycemia that triggers the potassium, that low glucose. It enters through GLUT1, and this pathway is unknown. And then there is a potassium independent pathway where there is another. And when there is and where there is membrane repolarization via sodium and calcium channels, and then the calcium also enters into the cell, and then there is spike in intracellular calcium, and there is the release of glucagon by the exocytosis into the blood circulation. So you can see the difference between the high glucose response and when there is low glucose response, and how the blood sugar levels is being maintained. So here, while the pancreas, it's the primary source, and it is not the only one. Glucagon is not only synthesized by the pancreas, but also by the secondary sources and the tertiary sources. And here you can see that the primary sources alpha cells in the islets of Langerhans, the secondary sources L cells and intestinal endocrine cells of the small and large intestine. And another source is that glucagon is synthesized in the brain and the salivary glands, and then it gets into the bloodstream where it reaches the liver, adipose tissue, and the skeletal muscles. And in the liver, the primary target is to maintain the blood glucose levels. and adipose tissue just targeted for lipolysis. And skeletal muscle, this is the critical exception where the muscle lacks the glucagon receptors and muscle protein is preserved during glucagon spikes, and muscle glycogenolysis only occurs in response to epinephrine during stress. So this is the slide which shows the synthesis and sources of the glucagon. Now the mechanism of action, that is glucagon G protein coupled receptors cascade. And glucagon operates via the classical G protein coupled receptor complex cascade. And when the glucagon binds with the G protein receptor on the hepatocytes, inactive, and it activates the adenylate cyclase, which converts ATP into the cyclic messenger, that is cyclic AMP. All right, so mm, then there is amplification, and there's adenyl cyclase converts ATP into secondary messenger, that is cyclic AMP, and then rising cyclic AMP levels activate the cyclic AMP dependent protein kinase A, and with the protein kinase there is phosphorylation, that which is the key regulatory downstream enzymes and activate the catabolic processes and inhibit the anabolic processes. So this is how this glucagon it acts via the GPCR cascade, and glucagon also activates the IP3 pathway and increases the intracellular calcium. Okay, so in this slide, you can see there's that in the liver, glucagon is the master of glucose production, you all know, and it activates the cyclic AMP cascade to convert phosphorylase B into in its active form, all right? And and degrade the glycogen into glucose 1 phosphate, and then it increases the dephosphorylate and released into the bloodstream as free glucose. So for proteins, it stimulates increased amino acid uptake by the liver by the gluconeogenesis, and it inhibits the hepatic protein synthesis to degrade the hepatic protein. And this is the rate limiting step where it accelerates the activation of enzyme converting pyruvate to phosphoenol pyruvate. And you can also see importantly that the blood amino acid levels stay stable because the muscle protein remain untouched. All right, so this is the carbohydrates and protein uh, metabolism on which the glucagon acts. And so we have learned about the glycogen metabolism and protein metabolism. Now this is the slide showing lipid metabolism, and adipose tissue. Glucagon activates hormone sensitive lipase, breaking down the triglycerides into free fatty acids, and inhibits new triglyceride synthesis by inhibiting acetyl-CoA carboxylase. And in the liver, when the free fatty acid released into the blood, they go into the liver for the ketogenesis and enhances CPT1 activity by decreased malonyl-CoA, and then the fatty acid undergo detoxidation to acetyl-CoA, which is converted to ketone bodies. And here you can see that the acetyl-CoA is forming acetoacetate, and that forms as 3 beta hydroxybutyrate, which is the ketone bodies. And then it is released into the blood after the liver, and blood levels of fatty acids and ketone rises, providing alternative fuel for peripheral tissues. All right, and So this is how the regulation of glucagon, it's a feedback loop, and where we can see the regulation which is balanced. This is the stimulate. These are the stimulators, and these are the inhibitors. And stimulators are the gas pedal, and the inhibitors are the brakes. Okay. So hypoglycemia is the primary trigger which stimulates the glucagon, amino acids, which arginine and alanine are dominant, and they stimulates the release of glucagon. Fasting and exercise, in which we experience hypoglycemia. Then GI hormones like cholecystokinin and gastrin, they stimulate the glucagon release, and vagal activity like acetylcholine, and sympathetic activity, that is beta-adrenergic stimulation, epinephrine and norepinephrine. They are the stimulators of glucagon release, while the inhibitors are glucose, that is when the blood glucose levels become high in the state of hyperglycemia. So this is the inhibitor of the glucagon, and insulin. It has got the direct negative feedback, and glucagon stimulates the beta cells to release insulin, which then inhibits glucagon. And this is a very interesting fact. And somatostatin is the paracrine inhibitor, GI hormone in which secretin and GLP-1. That is, GLP-1 is the GLP-1 and free fatty acid and keto acids, neural alpha-adrenergic stimulations. They are the inhibitors of the glucagon. Um, Clinically, glucagon is a lifesaver in severe hypoglycemia or insulin shock, and it is the primary indication to use glucagon in severe hypoglycemia, and it rapidly reverses insulin shock in an emergency state. Um, and it is also used in the heart failure and beta blocker toxicity when the patient ingests an abundant amount of beta blocker. So, in the state of heart failure, glucagon exerts a direct positive inotropic effect on the heart, that is increased force of contraction, and the mechanism of action is the increased calcium entry into the cell via cyclic AMP and entirely bypassing blocked beta-adrenergic receptors. And this glucagon is advantage over norepinephrine as it does not produce arrhythmias, tachycardia, or increase oxygen consumption, improve renal perfusion. Okay, and it is also used in acute pancreatitis and in radiological procedures, as it relaxes the smooth muscle and exerts an inhibitory effect on the exocrine secretion of the pancreas. So basically, the glucagon is the catabolic driver, and it is produced by alpha cells to prevent hypoglycemia, and it acts on the liver and the fat, but never in the muscles. And somatostatin is the paracrine protector that keeps the entire system from overshooting. And understand these pathways, and you will understand the foundation of the metabolic medicine. And So, in this slide, you can see that somatostatin is the universal brake, and the chemistry is always already being discussed in the previous slide. And you can see that it is secreted in response to food intake factors, that is, increased blood glucose, increased amino acids, and free fatty acids. And this is also the telecrine action inhibiting anterior pituitary growth hormone release, which we have already discussed in the growth hormone lecture. And you know that the somatostatin is secreted by the D cells and regulates nutritional influx by inhibiting gastrin, cholecystokinin, gastric inhibitory peptides, uh, motilin, gastric acid, and gallbladder contraction. and it is in the pancreas. It is secreted by the delta cell, and it has an intra neuromodulator, strongly inhibiting the secretion of both insulin and glucagon. So they are the clinical tactics of somatostatin, and it suppresses the excess growth hormone secretion from pituitary adenomas in the case of acromegaly, whereas it dampens the dangerous hypersecretion of systemic GI hormones in the case of gastroenteropancreatic neuroendocrine tumors, and it causes profound splanchnic vasoconstriction, safely reducing the portal hypertension to stop bleeding in the cases of esophageal varices. That happens in the cirrhosis of the liver, as you have already studied, maybe, and it directly inhibits the secretion of bicarbonate and digestive enzyme from the exocrine pancreas, allowing surgical sites to heal dry in the cases of pancreatic fistulas and postoperative leaks. Okay, so in this picture, you can see the synthesis of insulin and glucagon in fed and fasting state, and here the fed state that is from 0 to 120 minutes post meal, and that was triggered by the plasma glucose spike and amino acid and free fatty acids. And you can see the hormonal shift that's filling surge by the beta cells, and glucagon is actively suppressed, and somatostatin releases to modulate the nutrient influx, and glycogenesis and lipogenesis dominate, and glucose is cleared from the blood into the muscles. All right, so and the fasting state that is after 120 minutes post meal, and the plasma glucose drops, and um, there is, you can see the glucagon surge by the alpha cells, and insulin levels become baseline, and metabolic results are glycogenolysis, gluconeogenesis, ketogenesis dominate, and the liver pumps glucose and ketones into the blood for the brain and peripheral tissue. They're the key takeaways. They're high yield reviews that insulin promotes energy storage, that is anabolic action, and glucagon prevents hypoglycemia by mobilizing energy via uh, GPCR, cyclic AMP, PK pathways. Tissue specificity, that glucagon acts primarily on the liver, while the glycogenolysis and gluconeogenesis and adipose tissue exhibit lipolysis and ketogenesis. And skeletal muscle lack glucagon receptor, and somatostatin is the universal brake and is a potent paracrine and endocrine inhibitor, and its synthetic analogs are therapeutically vital for managing GI hormone, and gastrointestinal adenomas, bleeding, esophageal varices, and pancreatic fistulas. So I hope it makes sense, and if you have any queries, you can contact me. And I hope that you understand all these slides. Thank you so much.